All right, welcome. Today we are tackling carbohydrates, but I want you to think about them in a whole new way, not just as something you eat. We're going to treat them as life's master blueprint, a set of architectural plans that can build structures for both immediate energy and incredible, long-lasting strength. I mean, think about it. On one hand, you've got that quick energy burst you get from bread or sugar, and on the other, you have the tough, rigid structure of a tree's cell wall. It's kind of wild that both are built from the same family of molecules, carbohydrates. So, that leads us to the big question, doesn't it? How can one type of molecule be so incredibly versatile? Well, the answer isn't some kind of biological magic. It's actually a really beautiful principle of chemistry. A molecule's function comes directly from its structure. So if we want to understand how carbs can do so much, we need to unfold their molecular blueprint. Okay, so here's our game plan. We'll start with the why these sugars are so essential. Then we'll get into the actual chemical blueprint. We'll meet the simplest building blocks, learn how to draw them, and then most importantly, see why their shape is literally everything. And of course, we'll wrap up with a quick review for your exams. First things first, let's talk about why we should even care. Before we get lost in the atomic details, let's appreciate the bigger picture. Why does life depend so heavily on these molecules? Well, their job description is huge. They're the main fuel that powers almost everything our cells do. They're the structural backbone for things like plants. They get stored away as a rainy day food source. And they even act as these little molecular name tags on our cells, which is absolutely crucial for communication. So yeah, from the energy in your body to the structure of the entire biosphere, carbohydrates are, without a doubt, essential players. Okay, now for the blueprint itself. What actually makes something a carbohydrate? Let's get into the fundamental chemical rules and the core materials that define them. So that definition, polyhydroxyaldehydes or ketones, sounds a bit intimidating, but let's just break it down. It's really systematic. First, polyhydroxy just means many hydroxyls, those OH groups, so these molecules are basically decorated with them. And second, every carbohydrate has either an aldehyde or a ketone functional group. That's its reactive center. It's this 1-2 punch, the multiple hydroxyl groups plus that special carbonyl group that gives them all their unique properties. And this general formula is actually where the name carbohydrate comes from. It looks like for every carbon atom, you've got the equivalent of a water molecule, a hydrate of carbon. Now, this is a really handy starting point for a lot of simple sugars, but just keep in the back of your mind that it's more of a guideline than a strict, unbreakable rule. Some of the more complex carbs can get a little creative with it. So, just like any good architect, we need to sort our materials. We're going to start with the most basic building blocks, the individual bricks, if you will, which are called monosaccharides. We basically group carbohydrates by size. You've got monosaccharides, which are just single sugar units. Then you have oligosaccharides, which are short chains of a few sugars linked together. And finally, polysaccharides, which are these massive polymers made of hundreds or even thousands of units. For today, though, our focus is going to be squarely on the little guys, the monosaccharides. Now, even within the monosaccharides, there's a huge design choice right at the beginning. It all comes down to where that special double-bonded oxygen, the carbonyl group, is located. If it's sitting at the very end of the carbon chain, it's an aldehyde, and we call that sugar an aldose. If it's tucked away anywhere else in the middle of the chain, it's a ketone, and we call it a ketose. Simple as that. And here's where it gets really powerful. We can combine these two labels, its functional group and its size. For example, ribose is a 5-carbon aldose and aldopentose, and it's the literal backbone of RNA. Glucose, a 6-carbon aldose and aldohexose, is our body's primary fuel. And its cousin, fructose, a 6-carbon ketohexose, is the sugar we find in fruit. See, this simple little grid helps us organize a whole universe of incredibly important molecules. Okay, so we've got our building blocks sorted. Now, how do we actually draw them? This is super important. We're going to look at 2 key ways to represent these 3D molecules on a 2D page: the Fischer projection and the more realistic Haworth projection. First, let's talk about D and L configuration. It's a simple but very specific rule. First, you find all the chiral carbons. Those are the carbons attached to 4 totally different things, okay? Now, out of all of them, find the one that's farthest away from the carbonyl group at the top. The final step is just to look at the hydroxyl group on that one specific carbon. If it's pointing to the right, it's a D sugar. If it's pointing to the left, it's an L sugar. And a fun fact, pretty much all the sugars that matter in biology are in the D form. And check this out. This is where that simple D/L rule becomes so powerful. You can see here how the entire family of D-aldoses is built up systematically, all starting from that one simple block, D-glyceraldehyde. Every time we add another chiral carbon, the path splits in two, doubling our possibilities. It's this logical progression that generates all the incredible structural diversity we see in nature. Now, the flat drawing on the left, the Fischer projection, it's a great 2D blueprint, but it's not the whole story, not by a long shot. In reality, in a solution like water, sugars with 5 or 6 carbons don't just stay in a straight line. They actually curl up and form stable rings. And to draw that, we use the much more accurate 3D model you see on the right, the Haworth projection. So how does this even happen? Well, it's a really clever bit of internal chemistry. That linear chain is super flexible, and the hydroxyl group on carbon number 5 can just swing around and attack the aldehyde carbon at the top, which is a bit electron poor. When that happens, they form a new bond, creating something called a hemiacetal, and locking the molecule into a much, much more stable ring structure. And this is the form glucose is usually in inside our bodies. So we've seen all these tiny changes in a blueprint. A group gets flipped here, a ring forms there. But do these little details really matter? Oh, you bet they do. Let's connect these architectural details to their absolutely massive impact in biology. And the key concept here is something called stereoisomerism. Stereoisomers are fascinating. They are molecules that have the exact same atoms connected in the exact same order. The only difference is how those atoms are arranged in 3D space. It's just like your left hand and your right hand, same parts, but mirror images. This slide is the perfect example. Look at glucose and galactose. They're stereoisomers, and they only differ at one single position, the hydroxyl group on carbon number 4. That's it. We call pairs like this epimers. But that one tiny tweak in their 3D shape is enough for our enzymes to tell them apart completely, which leads to totally different metabolic pathways. In biology, tiny architectural details have huge, huge consequences. Okay, let's pull it all together. Here's a rapid-fire review of the most critical concepts we've covered. This is your high-yield summary to really nail down the fundamentals of carbohydrate architecture. All right, let's run through the essential vocabulary. You absolutely need to know what a carbohydrate is. You should be able to spot a monosaccharide and classify it as an aldose or ketose. And crucially, make sure you understand the difference between a general isomer and the more specific stereoisomer, and know when to use a Fischer versus a Haworth projection to draw them. If you take just one thing away from our talk today, let it be this. All the incredible complexity we see with carbohydrates, from energy fuel to structural beams, all of it comes from just a few simple, elegant rules of chemical design. It's a really beautiful example of how nature builds amazing complexity from very simple beginnings. We've seen just how powerful these single sugar units are, but the story definitely doesn't end here. The real magic starts to happen when these monosaccharides link together. So the question I want to leave you with is this. How do these simple sugars join forces to build the massive, complex carbohydrates that literally shape our world? We'll dig into that next time.